Testing for cancer inside the body using circulating nanosensors, as we discussed last time, is a very promising approach that may eventually allow us to screen for cancer in a way that's completely non-invasive. But there are limitations. First, as you're aware, anytime a substance is being developed for use in a human, there are significant regulatory hurdles that must be dealt with. The FDA protects all of us by having a very rigorous process in place to ensure that new medical procedures or treatments are tested thoroughly on human patients before they become accepted practice. Second, not all cancers respond equally well to testing within the body. In particular, we talked about the fact that in vivo imaging approaches can only really catch solid tumors. A leukemia, for example, is a blood cancer, and it doesn't have as many malignant cells that stand still. Same thing goes for tumors that are metastasizing, where there are cancer cells traveling through the bloodstream. The cells that are leaving the primary tumor are moving around, and before they colonize as a metastatic tumor, they wouldn't show up by imaging. So let's zoom in on some different approaches that may be usable outside of the body, and that are especially relevant in the short term. These are the approaches that may be relevant when we want to analyze cells or cancer markers in the blood. It's also quite possible that we will see nanomaterials appearing sooner as cancer diagnostic tools when they're used in vitro or outside the human body, given the lower regulatory burden of getting these things into clinical practice. Many of the tests currently used for cancer screening look for blood-borne markers, and they're performed with this type of in vitro or test tube approach. For example, the PSA test that is used for prostate cancer screening is performed using blood samples drawn from patients. The PSA test measures a protein that is released from the prostate if a tumor is creating pressure within the organ. This in vitro test, and most others actually, are not accurate or sensitive enough to catch cancer early. So there's a real need for improved testing systems. Now, the key feature that a testing strategy has to have to be effective for early cancer detection is a very high level of sensitivity. Now, what do I mean by high sensitivity? So if we think about what's going on in this sample that's going to be taken from a patient, the molecules that can be used as markers of cancer and other diseases are going to be present at levels that are correlated with the burden of the disease or the size of a tumor. If a tumor is very small and still localized, it will not release many molecular markers into the bloodstream. Or if we're thinking about diagnosing a blood cancer, like one that affects the white cells found in blood, the cancer cells will be a tiny population compared to the red blood cells. That means that if we're going to take a sample of blood and use it for cancer screening, whatever method we use must be able to track very low levels of a targeted molecule in the presence of a background of an excess of cells and molecules that can't interfere with the detection strategy. Nanomaterials show great promise for achieving this type of sensitivity. As we've touched on before, the fact that nanoparticles are almost the same size as proteins or nucleic acid molecules means that there's the potential for each particle to only need one molecule to make it give off a signal. This is actually an area that my own research group works in at University of Toronto. And so I'll take the opportunity today to tell you about some of our work. And this is work that is focused on developing new types of nanosensors, and in particular nanosensors that will assist with early cancer diagnosis. And at the end, I'll also touch on another approach developed in another lab that's also extremely promising. So we got started in the area of developing new cancer diagnostics about 10 years ago. I first started my own laboratories at Boston College, and very soon after I got my lab going, I was offered a grant from the National Cancer Institute. This is part of the National Institute of Health in the United States, and I was asked to start up a project that would develop a new cancer diagnostic system. The NCI liked some of the preliminary results we had, and they thought the approaches we were pursuing, all of which relied on nanomaterials, had significant promise. Now, 10 years later, we have a very powerful chip-based system up and running that uses nanomaterials to very sensitively detect DNA sequences that are the markers of cancer. Let me tell you a bit about how this works. The chip we developed is a device that is made using the same processes that are used in consumer electronics application. We print chips at the wafer level, meaning that we can make thousands at a time. And this is really important if we eventually want to mass produce the devices we're developing for clinical use. I have one of these chips with me and we can take a closer look at it. Now, as you can see, this chip is tiny. Looks like it couldn't do much of anything, but as I'll tell you, it's very powerful. This chip allows us to monitor a whole panel of markers at once, and it does it with a very high level of sensitivity that lets us go directly into a clinical sample and see what's there. Now, the underlying substrate of this chip is silicon. And then if you were able to zoom on this and see what's here, you'd see a gold pattern. And this gold pattern is generated using a process called photolithography. You heard about photolithography when Ted was telling you about information technology. Photolithography, as you know, is the main tool that's used to pattern metals and other materials on chips. It uses light to define the areas where patterning should take place. We don't need this particular capability to make our chips, but there are now types of photolithography that can be used to make nanostructures. So this is a huge tool in nanotechnology, and the resolution of photolithography gets better every day. Now, back to the layout of our chip. So rather than making you squint at that tiny chip, we can look at a drawing so you can see the features more clearly. The yellow squares that you can see, those are made out of gold, and that's where we're going to contact the chip to read out the signal. The pointy things that you see are leads that separate the sensors from the contact, so we don't get any short circuiting. We put our sensors at the ends of the leads. Now, the printing of this chip on a silicon wafer is not what gives us our nanoscale features. Instead, it's what we actually do once we bring the chip in the lab. So the chips get fabricated at a semiconductor fabrication facility, and then they come back to us and we post-process them. So if you look at the surface of our chip, you wouldn't be able to see this by eye. But it turns out that there are tiny holes in the surface of the chip, and they were put there purposely to provide a place for us to introduce nanomaterials. We take our chips, we put them in a bath of metal salt, we apply a potential, and that causes metal to electroplate onto the chip. And this is what gives us our nanostructuring. As the metal plates onto the chip, it forms tiny nanostructures that are actually quite easy to manipulate. So, for example, if we use 3 different conditions for electroplating, we can get 3 different levels of nanostructuring. So you're looking at images that are collected using scanning electron microscopy, and these are 3 different sensors that we made, and they were all made side by side on our chip. The one that is on the left has no nanostructuring. It's basically smooth. While the one in the middle has pretty large nanostructures that are about 200 nanometers. Now, the sensor on the right has very fine nanostructures that are about 20 nanometers. So why do these different levels of nanostructuring appear? We actually use the same materials to make them, same conditions in solution, except for one condition, and that has to do with how fast we do the electroplating process. If we make it go very fast, the metal plates onto the chip very chaotically, and the voids formed in the sensors don't get filled in. If we go slowly, we don't get any nanostructures because all of the voids are filled. So you get this nice, smooth sensor. So this is a nice, easy way to control nanostructuring. Now, how can we use these to detect biomolecules, and what role do the nanostructures that we've created play? So we can design and synthesize a DNA sequence that is complementary to whatever we want to detect. We know about the genetics of cancer cells. We know about the genetics of pathogenic organisms where this approach might also be valuable. And so we can identify that sequence, program it into our DNA synthesizer, and then attach it to our sensor surface. When the sequence that we're looking for is present in a sample, it binds to the sensor through the type of hydrogen bonding and base pairing that we talked about a few lectures ago. This binding event causes an electrical current to be given off, and that's what we're able to measure. We can put a variety of sensors on one chip and collect a profile of signals that can tell us about the type of cancer cell present in a sample. Now, it turns out that the presence of nanostructures on our sensors has a dramatic effect on the sensitivity of the approach. If we use a smooth sensor and we have a solution that contains a known number of DNA molecules, we've been able to determine that we need at least 1 million molecules in a sample in order to get that sensor to give off a response. So 1 million molecules, that's quite a large number. That's not typically what you have to work with in a clinical sample, so we obviously need to push past there. So now if we take our sensor with 200 nanometer nanostructures, our sensitivity gets better, but we still need hundreds of thousands of molecules present to get an accurate reading. Now, when we move over to the sensor that has the 20 nanometer structures, all of a sudden we only need 100 molecules in solution to trigger detection. This is a huge improvement over state-of-the-art that allows us to contemplate using these sensors to analyze clinical samples for cancer biomarkers. So when we studied these different levels of nanostructuring and we were able to pull out this trend and show that the nanostructuring gave us much better sensitivity, we were very excited about making that discovery. So why does this work so well, and why does nanostructuring matter? One thing that we discovered in the course of our work is that the closer a nanostructure is in size to the molecules that are being looked at for detection, the better the sensors work. So now if we think about what's going on at the nanoscale, the molecules that are immobilized on a sensor that are being used as molecular bait, they're trying to reach out to find their complementary sequences in the sample. Now, if they're on a surface that is flat, on their size scale, they aren't really accessible. It's kind of a sea of bait molecules, and they can't really see over their neighbors. Now, if instead they're displayed on a small nanostructure, one that has a radius of curvature that they can feel, they're much better at finding the molecule they're trying to detect. The nanostructuring, and again this size matching, creates room around the DNA strand trying to see the molecule that it's looking for. So it's a bit like being in a movie theater. If you're in an old theater that has the seats arrayed on a flat floor and you're vertically challenged the way that I am, as soon as somebody sits down in front of you, your vision is obscured and you can't see what's going on. But if you go to one of these nice new theaters that has the tiering, doesn't matter how short or tall you are or how short or tall the person in front of you is, you have an unobstructed view and you're able to see what's going on in the entire room. But if the seats in that theater were only tiered by an inch, that wouldn't help, right? The spacing of the seats need to be approximately on the human size scale to make a difference. Same thing with our DNA strands. The nanostructuring needs to be on the order of the molecular size scale to give them some room to see what's in the solution. Now, let me tell you about a specific study we did using this chip. There is a certain form of leukemia, chronic myeloid leukemia, that affects white blood cells. Specifically, it affects the granulocytes that are important for fighting infection. It's one of the most treatable cancers, with very effective drugs available to arrest it. However, it's very important that it's diagnosed and classified very specifically so that the right drug is used to treat it. There are many different forms of leukemia, and it's really just this certain form that will respond to the drugs that I'm mentioning. So the origin of this disease is very interesting. It arises because a specific break occurs on chromosomes 9 and 22, and then they recombine to form a fusion. This creates a new DNA sequence that is not present in healthy cells. As a result of the sequence being present, a mutant protein is produced that causes the cell to be overactive and divide too quickly. This is what increases the level of white blood cells to a level that the body can't sustain. Now, we don't know why this chromosome break occurs, but there have been suggestions that it could result from exposure to radiation. There must be other causes, though, because there are people affected with this type of leukemia that we know haven't been exposed to radiation, so that's still a bit of a mystery. The good news is that a drug is available that very specifically targets this protein and shuts the mutant cells down. Since the mutant protein is only present in the leukemia cells, the drug has no effect on other cells and has few side effects. And there are very few drugs on the market that are this specific. So now let's talk a bit about how this type of cancer is diagnosed presently. So if someone has a high white cell count, a sample of their blood is drawn and then it's taken to a lab. Once it's at the lab, a research technician will take the sample, they'll isolate all of the DNA from the blood, and then they'll start analyzing it. Now, even from a large sample of blood, that 10 mils that usually goes into a vacutainer, there's not enough DNA to analyze using most methods that are available in clinical laboratories. So a process called PCR, the polymerase chain reaction, is used to amplify the CML gene. PCR makes many copies of a DNA molecule. So just like a Xerox machine, it just runs lots and lots of copies off of a single original. And it can produce billions of copies from a single original molecule. Now, once you have this much DNA in a sample, it's much easier to use pretty simple methods, for example, monitoring the emission of light coming from a DNA binding dye, to analyze the sample. In the figure that you're looking at, the vials are showing you the strong fluorescence signal that's obtained once a CML sequence is amplified. So this works, but it's expensive, it's slow, and it can only be done in specialized laboratories with very experienced laboratory technicians running the tests. And so that's why we decided to test whether or not our microchip-based system could be used as a substitute. So here, instead of amplifying the thing that we're trying to detect by running off copies of it, we're trying to turn up the volume of the signal that it produces. Seems to us that it's more straightforward to do detection that way. So to test our chip-based approach for leukemia detection, we designed a chip that would display a DNA sequence specific to the CML gene fusion on a nanostructured sensor. Recent advances in the synthesis of artificial DNA make it possible for us to have a machine in the lab that is able to make any DNA sequence we need. Once the DNA sequence is made and ready, we attach it to the sensor by putting a sulfur atom at its end. We touched on the fact before that sulfurs are very good at sticking to the metals that make up our sensor. So that allows us to take a specific sequence and anchor it to our chip. So then, once the chip is ready for a measurement, we calibrate it, and then we introduce a sample containing an unknown number of cancer cells. Data is produced from the chip that allows us to determine how high the levels of CML cells in the sample are. What you're looking at is raw data generated from our instrument that reads the chip, and it shows how much current is generated in the presence of varying numbers of cancer cells. We were able to do this kind of measurement both with samples taken from patients where the white blood cells were isolated and also in whole blood samples. The fact that we could get these kinds of readings in whole blood presents the possibility that maybe someday, rather than having to take a 10 ml blood draw from a patient, we could just do a pinprick and take a tiny drop of blood and analyze it, something that could be done right in a doctor's office. So we're very excited about the potential of this technology. What will it take to put this on the market and make it available to the clinical community? Well, we have a prototype instrument that we're using to validate the sensing approach. And I've brought a prototype of this instrument with me to let you take a look. So the way that this will eventually work is that our chip will be embedded in a cartridge that a physician can inject a sample into. So if you were able to see this up close, you would see the contacts of the chip sticking out here. So the chip is embedded in the cartridge. And these foil packs, those are what hold our reagents. And so those are the things that we need to be able to run this assay. And what will happen is that the sample gets introduced into this cartridge. The cartridge then gets slid into the instrument, locks in, and then all one has to do to start the analysis is to press one button on the instrument, and then within 5 to 10 minutes, an answer will appear to indicate whether or not there are cancer cells in the sample. So why are we able to make such a small testing unit? Why is this possible? Well, all of the signals that come off of our chip are collected electronically. So we don't need any optics in this instrument. We don't need any type of fancy detectors or excitation sources. We just need a pretty simple set of circuits that can apply potentials to our leads and then collect the signals from the sensor. So inside of this box is just a collection of very simple electronic parts. And so we think that this is an instrument that can be produced very inexpensively and put at a price point where every physician could have one in their office. Now, very often when I tell people about this technology, they tell me that it sounds like a Star Trek tricorder and it's too good to be true. But this type of approach, where information on species or molecules in blood can be reported very quickly, is slowly but surely making its way into clinical medicine. Take, for example, the way that diabetics can now manage their illness with handheld monitors that report on blood glucose levels. These are very simple instruments that are quite inexpensive. Now, the premise here is the same. We use the same guts in our instrument that you find in a handheld glucose monitor. But our analytes are much scarcer in the samples that we analyze, hence the need for nanomaterials. But there is every reason to believe that disease diagnosis will be made much faster, cheaper, and straightforward in the next decade, and nanomaterials will play a big role in this paradigm shift. Now, let's look at another advance in cancer diagnosis and management made possible by nanoscale sensors. We've discussed gold nanoparticles as good sensors to report on genetic disease and also the presence of hormones that can confirm pregnancy. These materials, it turns out, are also very powerful for cancer detection. They can detect proteins in the same way that we discussed for the case of determining whether a woman is pregnant. The red color that a gold nanoparticle produces can be concentrated on a chip. And the fact that these red absorbing nanoparticles are nanoscale makes them much easier to handle. A recent study using gold nanoparticles to do protein detection looked at detecting PSA, the protein I mentioned at the beginning of the lecture that is a marker for prostate cancer. And they used a gold nanoparticle approach rather than conventional methods that are currently used. The commercially available PSA test is good at detecting large changes in levels of this protein, but it's not as good at looking at small variations. It's also not good at looking at very low levels of the protein. Men that are treated for prostate cancer with a prostatectomy have trace levels of PSA in their blood post-operatively. And so the levels of PSA go way down and they're actually not measurable using the existing test. And so why does this matter? Well, it turns out that how fast the PSA comes back after the operation can tell a physician quite a bit about what treatment regimen to use. And this decision should be made just when the PSA is starting to creep back up, not once it's already kind of on a steep trajectory. And again, the methods that are currently used for PSA screening simply can't look at these low levels that are present. So a study published in 2009 showed that the very sensitive gold nanoparticle approach was a great solution, and it could predict recurrence very quickly and accurately. So in this study, again done using gold nanoparticles, PSA detection data was collected for a series of patients, a pretty large cohort of patients. The data sets that were collected showed a dramatic difference in PSA reappearance in different patients. For some patients, the PSA stayed nice and low after the operation was complete. And this must have given this subset of patients really great peace of mind. For other patients, however, about a year after they had their prostate removed, their PSA levels started to creep up again. Now, there are many treatment options that can be helpful for patients that surgery doesn't cure. Here, with access to this technology, the oncologist treating these patients had the information that the cancer was not gone many months before they would have known using the conventional diagnostic assay. So it's clear that adoption of this approach should improve treatment outcomes for prostate cancer patients significantly. So we've looked at two approaches to in vitro cancer diagnosis that have significant promise. While in vivo approaches may help us do whole body screens for early tumors, in vitro approaches are probably also here to stay as they provide information on cancer cells and markers in the blood. But before we leave this topic, let's take a step back and think about these two different approaches and how they may be complementary and potentially used at the same time to more effectively manage cancer. So we have the in vivo approach, where we send the nanosensors into the patient, let them circulate, let them take advantage of the leaky vasculature of tumors, let them accumulate in tumors, and then we get a snapshot of a patient's profile that tells us where there may be an accumulation of cancer cells. So this is a very powerful approach. Again, it has the promise of detecting tumors when they're at a very early stage, and it should be a very simple measurement. We just need to look for light shining through the tissue and skin. So that is an approach that will continue to advance. As we mentioned when we talked about that material, that the safety of the imaging agents needs to be assessed. The regulatory burden on that type of approach is higher. So we then turn to our in vitro methods to look at how nanomaterials can be leveraged to look at samples that are taken out of patients. And we looked at two different systems. One that is a chip-based system that allows cancer-related nucleic acids to be analyzed, and another, a gold nanoparticle system that's very good at looking at low levels of protein. So here, one of the unique advantages of doing things on a sample, doing things on a chip or with particles, is that we can look for many markers at once. If we do things in vivo, we're usually looking for one marker. If we do things in vitro, we can look for panels. So again, these approaches are very complementary, and eventually they'll probably both be available and used together for more specific diagnoses and earlier diagnoses. So in the next lecture, we're going to switch gears and we'll start talking about nanomaterials as drugs. That's right, nanotechnology not only has the possibility of detecting medical problems, but it may also be just as useful in treating medical problems. So we'll get away of thinking about how you find trace analytes and samples or in the human body, and we'll really start focusing on the solution and new treatments for cancer that are based on next generation nanomaterials. So next time we'll really hone in on how treatments can be developed to selectively eliminate harmful cells. See you then. If you're a science fiction fan or a Raquel Welch fan, I bet you've seen the movie Fantastic Voyage. This 1966 movie has a plot that involves the invention of a miniaturization technology that allows a submarine to be scaled down so small that it can zoom around the human body and repair problems like clogged arteries or blood clots. You may remember from the movie that the team goes into the submarine and it has a series of misadventures. It encounters turbulence in the heart and attacked by white blood cells. In the end, they're finally able to repair a brain blood clot and escape from the body through the tear duct. Now, this is clearly science fiction. We can't miniaturize people, and we probably never will be able to. Down at the nanoscale, a human-type body wouldn't even work because of the way we need to take things in from our environment. And yet, we'll see in this lecture how nanomaterials are being able to achieve something similar, but without the miniaturized people. You already know how gold nanoparticles can be modified to recognize specific molecules and diagnose disease. You also know about how semiconductor quantum dots can be used to seek out tumors in vivo by selectively accumulating in cancer cells. Now we'll look at how nanomaterials that seek disease cells in the body can also be used to destroy those disease cells, just like the microsubmarine in the Fantastic Voyage, except without the tiny people inside. Which is good news, actually. In the Fantastic Voyage, the miniaturized people were somehow able to breathe regular oxygen, even though one molecule of oxygen would have actually been too large to fit in their mouths. So things simplify quite a bit if we think about making tiny submarines to do these kinds of jobs, but ones that don't need a human crew on board. We'll see in this lecture 3 different ways that a familiar material, gold nanoparticles, can target and eliminate disease cells. Gold nanoparticles are really good at moving around the circulatory system, and just like quantum dots, they're small enough to move through even the smallest of capillaries. But how good they are at doing this actually depends on their size, which can range from about 1 nanometer to over 100 nanometers. A recent study compared 20 nanometer and 100 nanometer particles that were introduced into a mouse that carried a tumor. The animals were examined after different time intervals, and then portions of their tumors were analyzed. So as we look at gold nanoparticles that can selectively eliminate cells, we have to keep an eye on the sizes of the particles. Smaller seems to be better to enable the particles to travel away from a blood vessel and penetrate a tumor. This makes sense, as the particles need to be able to slip in between the cells that make up a blood vessel. Now, what properties do gold nanoparticles have that allow them to kill disease cells? We've discussed to some extent how these materials interact with light. For example, when we talked about the color changes that result depending on the spacing between particles. And in that case, the nanoparticle was emitting a signal that we were following. Here we'll be looking at a different phenomenon, the ability of gold nanoparticles to absorb light and then convert it into heat. If you think back to our discussion of quantum dots, you'll remember that when they're photoexcited, light goes into the particles and then the energy contained within comes back out in the form of emitted light. It turns out that the electronic structure of quantum dots makes them good at holding the energy in an excited state that can be emitted as photons. For gold nanoparticles, when light goes in, the energy actually comes back out as heat. These nanoparticles are not good at keeping the light in an excited state, and so the light is instead converted into vibrations that produce heat. And as we'll see, this effect can be used to kill cells that gold nanoparticles selectively enter. Now, why would a bit of heat kill a cell? Well, most biomolecules lose their shape when they're heated up, and this interferes with their ability to function. And in certain cases, gold nanoparticles get really close to molecules. For example, it was discovered several years ago that a 1.4 nanometer gold particle was exactly the right size to sit in the major groove of DNA. You'll remember that the double-stranded structure of DNA contains 2 grooves or spaces between the 2 strands of DNA. And these grooves are actually asymmetric, and the larger one, which is about 2.2 nanometers wide, is referred to as the major groove, while the smaller one, which is about 1.2 nanometers wide, is the minor groove. So if a 1.4 nanometer gold nanoparticle nestles itself into this major groove space and light causes the particle to give off heat, local heating is going to result. And that heat can cause the two strands to dissociate. And when DNA strands dissociate, well, that really interferes with a cell's ability to function properly. And roughly the same thing can happen with protein molecules. Certain proteins will even non-specifically attach themselves to gold nanoparticles, and these proteins can also be denatured when heat is produced. Really, all cellular structures, which are held together by non-covalent interactions like hydrogen bonds and van der Waals interactions, can be perturbed by heat. So exciting gold nanoparticles with light can basically turn a good detection sensor into a sensor that can both seek and destroy a cell. So let's take a look at an example of how this can work. A group at Georgia Tech has done some very nice work with gold nanoparticles that selectively enter and kill cancer cells. They were able to use a trick that we've already talked about in order to get the particles to recognize the cells. Basically, they attached an antibody to their gold nanoparticles. You'll remember that many types of cancer cells overexpress a protein called the epidermal growth factor. And there is an antibody to this protein that's available. So they attached that antibody to gold nanoparticles and verified that the nanoparticles were very specific for cancer cells. So to do this, they took two types of epithelial cells, one healthy and one cancerous, and they tested the activity of the gold nanoparticles. Epithelial cells are the kind of cells that line the surface of tissues, like skin, and cavities, like the mouth or the stomach. The healthy cells don't have much of the receptor that the particles were designed to bind to, while the cancer cells, which were derived from an oral cancer tumor, have high levels of this receptor. So if you look at this image, you can see the difference in gold nanoparticle binding to the two different types of cells. So on the left are the healthy epithelial cells, and on the right are the cancer cells. You can see that there are many more nanoparticles inside the cancer cells. Now, when these researchers irradiated these particle-containing cells with light, they saw much higher levels of cell death for the cancer cells. So this provided really good proof of principle because it showed the gold nanoparticles could be used to get targeted ablation of cancer cells. And it turns out that we can use the same approach to selectively kill bacterial cells. We can also get antibodies that are specific for certain types of bacteria and basically play the same game. So in a study done at the University of Arkansas, a group in the microbiology department there generated gold nanoparticles that carried an antibody specific to the bacterium Staphylococcus aureus. You've probably heard of the infections caused by this bug. They're typically referred to as staph infections. And they can be quite serious, even or especially if the bacteria ends up in the blood. So when these particles came into contact with the bacteria, here the nanoparticles didn't actually go inside the cell, but instead they just sat on the membrane. And when the particles were excited with a laser, they disrupted the cell and caused the bacterium to break open and die. And the images that you're looking at show this quite clearly. On the left, you can see a bacterium with the nanoparticles attached to its surface. And on the right, you can see how the cell wall has broken open. So the use of nanoparticles to seek and destroy certain cell types is applicable not only to human cells, but also to bacterial cells. And it's potentially a very powerful way to kill and target problematic cells while leaving other nearby cells unharmed. But so far, we haven't looked at whether this gold nanoparticle approach to cancer or bacterial cell elimination works in a living system. So why not? Well, here's the catch. Gold nanoparticles typically absorb in the 500 nanometer to 600 nanometer range. The larger the particle, the more that absorption shifts to a higher wavelength, or the red part of the spectrum. But in this part of the spectrum, there is lots of absorption by tissue, as we discussed in the previous lecture. So if the tissue all around is absorbing the light, that means that very little light will make it to the gold nanoparticles, even if we use a powerful light source like a laser. So while gold nanoparticles give us really important proof of concept for using nanomaterials to target and damage specific cell types, this absorption of light by surrounding tissue really appears to limit the use of gold, at least this kind of gold, in living systems. But let's take a look at a clever approach that circumvents this issue and uses gold in a slightly different way that solves this problem. It turns out that one way to solve the absorption problem is by making a modified type of gold-based nanoparticle. Several years ago, researchers at Rice University figured out that if instead of using nanoparticles entirely composed of gold, they instead used a core of another material, like silica, and then they coated it with a very thin layer of gold. And what they observed is that this shifted the absorption of the particle way into the infrared. And infrared is a much better region of the spectrum for getting light to pass through tissue. The absorption by tissue is really minimal in this part of the spectrum, so the light can actually reach the nanoparticle. So first, let's take a look at how these gold-plated nanoparticles are made. You start off with a particle composed of silica, which basically has the same composition as sand, but the particles are obviously much smaller than those that you find in sand. They then get coated with tiny particles of gold until the gold shell grows together. And what's really neat about these materials is that as the coating gets thicker, the absorption peak shifts from the more red part of the spectrum to the more blue part of the spectrum. This makes these materials very tunable. That is, the thicker the gold plating, the more the absorption peak shifts to lower wavelengths. These nanomaterials really illustrate why being able to control materials at the nanoscale is so important. So just by varying the amount of gold plating, we can really tailor their optical properties and their heat-absorbing properties to make them really effective in particular applications. Now here, in order to use these as better cell targeting and killing agents, we obviously want to focus in on the materials with the thinner coatings that will absorb light more in the red and the infrared part of the spectrum. And it was found that these gold nanoshells could be used in the same way as the original gold nanoparticles, but we'll see that that shift that was achieved in the absorption spectrum allowed them to then be studied in real tissues. A study done at the University of Texas Southwestern provided a very compelling example of an in vivo study showing how powerful these nanoshells are. In this study, a mouse model of prostate cancer was generated by implanting tumors that grow from human prostate cells. The tumors were allowed to grow until they were about 40 cubic millimeters, and this is about the size of a pencil eraser, just so you know how big it is. The mice then had the gold nanoshells injected through their tail veins. After about 18 hours had gone by, the researchers then irradiated the tumors with 800 nanometer light for about 3 minutes. And then they monitored the mice over 3 weeks and measured the tumors over the course of this time period. Now, in the control group of mice that they monitored that didn't get any treatment, the tumors tripled in size over this time. But the tumors in the treated group, the group that got the gold nanoshell therapy, they shrank so small that the researchers couldn't measure them by the end of the 3 weeks. This was a really striking result. Now, despite the success of this study and the proof of principle that they achieved for using gold nanoparticles in a tumor that was buried under a little bit of tissue, the authors of the study pointed out an important consideration in how this work should be moved forward. And it turns out that they got to the goal that they wanted to achieve. They were able to use the nanoshells to improve the absorption properties of the particles, and they were able to do a study in living animals. But they knew that there were still limitations. When they measured the penetration of infrared light into the tissue, they found that they could go a good distance, but once they went further than a centimeter, they had trouble getting the light into the tissue. So if you think about many types of tumors, like lung or breast tumors, these would need better penetration depths in order to be accessible. But there are solutions that may allow us to use these materials. One would be to deliver light to different places in the body directly, for example, by bringing a fiber optic bundle into the lung or another part of the body. So if we continue to run into trouble with getting light through tissue, there's always a way to solve that by bringing the light closer to the particles. But there's another way to think about this. Instead of using the type of light that we've been talking about, visible light, infrared light, we could think about using a different type of radiation that doesn't have these absorption problems. One of the most common methods of cancer treatment relies on what we call radiation therapy or radiotherapy. Here we're using radiation, but it's very high energy radiation, and it has much smaller wavelengths than the light we were just talking about. And these wavelengths of light can be used to directly damage cancer cells. We call this kind of radiation ionizing radiation because molecules, when they are hit with it, they're forced to give up electrons, and they become charged ionic molecules. And this triggers DNA to release electrons and causes it to self-destruct. In other words, ionizing radiation typically causes a cell to die. This type of external beam radiation therapy focuses a really powerful beam towards a tumor, and it's very effective at shrinking tumors. And the type of energy that we're talking about here is much easier to get through human tissue. So tumors that are buried under the skin can be treated this way. But in this approach, it's very difficult to cleanly target the tumor because there's always healthy cells in the vicinity. So what if we could use gold nanoparticles as an antenna for ionizing radiation? And we could use the fact that we can make them specific for cancer cells to help radiation therapy be more selective for tumor cells versus healthy cells. Now, how can gold or any other type of nanoparticle act as an antenna? Well, there's an effect called the dose enhancement effect that comes into play when you have radiation interacting with the junction between two different materials that have very different atomic numbers. And what we find in the human body are elements that mostly have pretty low atomic numbers. So carbon has an atomic number of 6, oxygen is 8, hydrogen is 1, and these are the common elements in cells. Gold, in contrast, has a very high atomic number, 79. So at the interface between the gold and the atoms of human cells, very high doses of radiation will be generated. And the enhancement in radiation dose can be as much as 500%. This is a really powerful effect. So we've seen before that gold nanoparticles can be directed at cancer cells, bind them and enter them selectively. And here potentially we could use the good penetration properties of ionizing radiation to more effectively get at these tumors that are buried under skin and tissue, but will leave the healthy cells untouched, hopefully. So let's take a look at a study where this idea was tested. A group of scientists at the University of Connecticut Health Center, collaborating with a company called Nanoprobes that is based in New York, were one of the first to explore how ionizing radiation could be used together with gold nanoparticles. They generated a mouse model of breast cancer, and they generated tumors within the mice that grew to about 50 cubic millimeters, about that eraser size again. They separated these mice into 4 different groups, one that didn't get any treatment, one that received radiation only, one that received only the gold treatment, and then one that got both gold and radiation. For the gold only and the no treatment groups, the tumors grew very quickly, and they enlarged by about 1,000% within 5 days. The radiation only group had slower growing tumors, but within 20 days, these mice had tumors that were actually identical to the control group. But amazingly, the tumors in the group of mice that had been treated with both the gold nanoparticles and radiation, the tumors were completely gone. They were too small to measure. And what was particularly neat about this study is that these researchers then followed the mice in each group for an entire year so that they could evaluate whether the animals that received the gold plus radiation treatment were healthy in the long term. And when they did, they observed an improvement in survival rate from 0% for the control group to 86% for the highest dose of gold nanoparticles. This is an amazing result. You know, if we were able to see the same type of effects in humans, cancer would be a much more treatable disease. Now let's take a look at a third way that nanomaterials can be used to selectively ablate cells. We've worked through a photothermal approach, where gold is used to generate heat, and an external radiation approach, where gold is used to amplify radiation coming from an external source. So I think you'll be able to appreciate this third approach. It turns out that gold nanoparticles can also be used as an internal source of radiation. This then eliminates the need to have radiation passing through healthy tissue at all. And so it's even cleaner than the amplifier approach we just spoke about. So first, let's talk through how internal radiation has traditionally been used without nanotechnology. And then we'll turn to how nanomaterials can improve the efficacy of this approach. The use of radioactive particles to shrink tumors is referred to as brachytherapy, from the Greek word for short distance. This approach is particularly effective for the treatment of prostate cancer. It is much less invasive than surgery and much less prone to complications, and so it's quite popular, as you can imagine, with patients. It can also be used to make surgery more straightforward by shrinking the tumor before a surgeon tries to excise it. So what's done here is that small gold seeds made of radioactive gold are implanted near a tumor. The gold is radioactive because it has an unstable nucleus that gives off gamma emission. The overall effect that results, the death of cells close to the particle, is the same as what's achieved with external radiation, but here it's much more localized and you don't have any healthy cells in the way. Now, one problem that can be encountered with this approach is that the radioactive seeds can be difficult to work with and somewhat heterogeneous with lots of variability in their sizes. Radioactive seeds of varying sizes are likely, less likely to focus on the tumor and more likely to focus elsewhere, perhaps causing inflammation, pain, and other side effects. So this is where nanotechnology comes in. As you now know, it's pretty straightforward to very precisely control the size of nanoparticles. So what people realized is that by using well-defined gold nanoparticles made of radioactive gold, they could greatly improve the reproducibility and reliability of brachytherapy. Let's look at a study where this idea was tested. At the University of Missouri, a group of clinicians and scientists got together and made radioactive gold nanoparticles to test their efficacy for brachytherapy. They were able to show that the gold nanoparticles had very little variation in their sizes and in the levels of radioactivity that they emitted. You're looking at a set of traces that illustrate how nanomaterials can be sized. A technique called dynamic light scattering is used to estimate the size of the particles and also to judge how much heterogeneity is present. The sizes of these particles were nicely clustered just around 100 nanometers, right around 85 nanometers, with very little variability. At most, the particles varied in size by about a factor of 5, from the lowest size observed to the highest size observed. And when conventional gold seeds are used, their sizes can vary by a factor of 100. So we've cut down the variability in size quite a bit by using nanoscale gold. So once this group knew that they had a good material to test, they made sure that the gold nanoparticles did not affect the blood of the mice injected with them, just to look at the side effects that might result. So they looked at the aggregation of platelets. They also checked that the hemoglobin levels didn't change, and this made it apparent that the gold nanoparticles didn't have any adverse effects on the mice. Then, using mice that had prostate cancer tumors, they tested their materials. And what they observed was that the tumors in the group of mice that were treated with the radioactive gold particles had a reduction in the volume of their tumors of about 82%, and no side effects like weight loss or hair loss. What's really exciting about this discovery is that it may present a way to effectively deal with tumor metastases. Usually, these tumor satellite sites are detected using imaging. And then depending on where they're located, external beam radiation is used to decrease their size. Small tumors, what are called micrometastases, are difficult to pick up with this type of imaging. so we usually don't even get the chance to treat them until they're larger. But with the approach I just described to you, the particles could actually find the tumors and shrink them, irrespective of size. And this would likely be a much more effective way to halt the progression of metastatic cancer. So now you know how nanomaterials can be used to seek and destroy cancer cells and other dangerous cell types. We talked about examples of cancer cells and also bacterial cells that can be destroyed using the same approach. For the cancer cells, we actually went through 3 different ways that we can use gold nanoparticles. We can get them to give off heat, we can get them to act as an antenna for radiation coming from outside of the body, or we can actually use them to deliver radioactivity directly inside the body. And in each of these cases, the ability to manipulate matter at the nanoscale was absolutely essential. So particles of particular sizes and with high levels of uniformity, this was critical in order to make these materials effective killers of cancer cells without harming other nearby cells. So the materials that we looked at may not be as smart as the microsubmarine in the Fantastic Voyage, but you can see that they certainly get the job done, and they would do it cheaply. So you may be thinking about the fact that the price of gold per ounce is pretty high, and you might wonder how much gold we would actually need to treat a patient. It turns out that it would probably just add up to about 10 milligrams of gold. So if someday the price of gold went up to $10,000 per ounce, we're still only talking about $3 per patient in terms of the cost of treatment. So that's one thing to keep in mind, that even when we're using expensive materials, if we're using them in their nanoscale form, it's not going to be cost prohibitive. So the work that I described on nanomaterials being used as drugs to kill cancer cells or bacterial cells is just beginning. And we talked about some of the challenges that still exist with getting light into tissue and really getting these materials to be effective. But it's clear that we're going to find solutions to make these materials work. And there's many other applications of the same types of approaches, including the treatment of heart disease, where destroying harmful plaques can be very important. So even though we don't yet have those nanoscale submarines, you can see that very powerful tools for medicine can be developed using pretty simple components. So in the next lecture, we will explore what you can think of as a division of labor in the mission to seek and destroy disease cells. Here we'll look at nanomaterials used as delivery agents for other drugs. And we'll find that this approach is actually farther ahead than the one we just discussed. Several drugs that are already approved by the FDA use nanodelivery systems, and they're already on the market and in clinical use. See you then. Have you ever thought about what happens to a pill once it lands in your stomach? Think, for example, about aspirin. In addition to curing your headache, it may also give you a stomachache. The way that we administer drugs is very often untargeted. And we introduce a drug and we let it distribute itself throughout the body. This can lead to a lot of side effects and irritation, like the aspirin-induced stomachache. So what we'll talk about today is how nanoscience may provide a solution allowing more targeted drug delivery. So what do I mean by that? Well, we know that nanomaterials can be targeted to certain types of tissues. We've looked at examples where that has worked for example, imaging cancer tumors. Now, if we could turn this paradigm around and learn how to use nanomaterials as drug capsules and target them to specific tissues, we could leverage this type of specificity for better treatments. This lecture will be the first of 2 focused on nanoscale drug delivery. And by that I mean that we're going to be focusing on systems that allow us to take a drug, package it up, and allow it to be delivered to the site within the body where it's going to exert its action. And we'll start by trying to understand the challenges of drug delivery And then we'll look at a variety of nanomaterials that can be used to meet those challenges. And we'll also discuss several of the leading anti-cancer drugs so that you're familiar with the agents that will be involved once we shift over to look how nanomaterials can be involved in their delivery. So that'll be this lecture. And then in the next lecture, we'll look at specific examples of nanopackaged drugs that are either on the market or in development. So by the time we get through all of this material, I think you'll understand how drug delivery is one of the areas where nanoscience is making a really significant near-term impact on medicine. So getting a drug to work in the human body is all about getting it to the right site at the right level. Now, why is this hard? So we almost always rely on means of distribution for a drug where the drug just goes all over the body. So if we take a drug orally in a tablet, like aspirin, it goes into the stomach, it's absorbed through the small intestine. We can also administer drugs through the eye or the ear or the nose. All of these places will take drug up into our system, but each of these are also delicate tissues that we don't want to perturb. And you might also be aware of the fact that in the last decade, we've become better and better at delivering drugs using patches. And so we put a patch containing the drug on the skin, and the drug then goes into the skin, but it does actually end up in the bloodstream. So this is not really a localized type of drug delivery, but something that's fairly delocalized. Now, maybe the one way that we're able to deliver a drug in a way that is very specific and localized are topical drugs. For example, if you get poison ivy, you don't take a pill, you put cream on your skin, and that works very well, and there are no side effects from that type of approach. But the skin is really one of the only places that we can do that. Anything that's affecting an internal organ obviously needs to be addressed from the inside. So for all of the drug delivery challenges, the main thing that is difficult is that the drug has to pass through a barrier. And this barrier may be our small intestine, or it may be the tissue that's in our ears or eyes or mouth. And the epidermis of our skin is just one example of a layer of epithelial cells. So our bodies have several kinds of these densely packed protective cells, and the drug has to be really good at getting through these barriers and into the bloodstream. In addition, many drugs just won't survive the acidic conditions of the stomach, and they get stuck in some of the other places that we mentioned. So in this case, we typically inject them right into the bloodstream. Now, this is obviously not a very convenient way to administer a drug. Nobody likes needles. But in some cases, it is the way to go because it gets more of the drug into circulation where it can start doing its thing. Now, what happens once a drug is in the bloodstream? Well, let's say we're injecting a drug into a vein, kind of the simplest case we can consider. That drug will first go into the heart and then over to the lungs along with the blood to get oxygenated. Once it picks up the oxygen, it'll then travel through the body in the arteries and then flow into the capillaries. And the capillaries are where the drug within the blood has the opportunity to make it into a tissue. So arteries and veins are designed for physical transport of blood, and really that's it. While it's the capillaries, the much smaller vessels, that handle chemical transport. And so they're really there to feed oxygen and other agents to tissues. So they have good exchange properties, whereas again, the veins and arteries don't. So it's the capillaries that will really allow drugs to travel into tissues. So how big are these capillaries? You probably have a picture in your mind of them being quite small, and we have to think about, are they so small that we can't really envision getting nanomaterials in them? But it turns out that they're about 10 microns, or 10,000 nanometers. So we don't really need to worry about nanoscale drug particles getting stuck. They'll be able to flow through just like the blood cells. Now, some of the toughest challenges in drug delivery is in the area of anti-cancer therapy. And tumors are some of the hardest places to reach in the body. They're very dense, and by that I don't necessarily mean that the cells are more closely packed, but they're much harder to penetrate, in part because the blood supply only reaches certain parts of a tumor. It really only comes to the outside. And then the cells inside can really be protected from the blood supply, which is how we're going to get drugs in. So that's a major challenge. And in addition, many of the drugs that are used to treat cancer have significant toxicity if they reach healthy cells. So it's quite important to try to engineer some type of targeting into their delivery. And we've touched on a feature of the blood supply around tumors that does help nanomaterials reach them. So if you remember when we were discussing tumor imaging, we went over the fact that blood vessels or capillaries can be quite leaky around the tumors. The leaks that are there actually create openings that measure about 500 nanometers. So this type of opening is too small for a red blood cell to leak out, but it's large enough for a drug packaged up with a nanomaterial to leak out if it's packaged properly and if the size scale of the particle is right. Okay, so that's how drugs travel around the body and into tumors. So the problem that needs to be solved here is how to more specifically target drugs. In the scenario that I just discussed, where we considered how a drug would be circulated by tagging along with blood, the drug will go to the lungs, it'll pass through the liver, it'll go through the heart and the kidneys, and there's lots of bad things that can happen to these organs while they're being exposed to drugs. And if, let's say, we're trying to target a drug to the pancreas, it's really unnecessary. If only we could send it right there and not have it stop in any of these other places. So what we'll find as we start to discuss nanomaterial-based solutions is that some nanomaterials can be very good at keeping drugs out of trouble in non-target areas. And we'll also find that nanomaterials can be helpful for drug delivery in a different way. Many drugs are difficult to formulate, and by that I mean that in order to take a compound and then administer it to a human, you have to have it either as a pill or in solution or in a patch in some type of formulation so that you can give it to the patient. And many drugs are really difficult to work with. If they're too hydrophobic, they won't dissolve in water. If they're not very good at making powders, you can't make them into pills. And so in certain cases, nanomaterials have really been able to solve this part of the drug delivery problem, which again is a formulation problem. And for drugs that get turned into intravenous drugs, one that we administer as a solution in a needle, it turns out that sometimes the solvents that are needed in order to take a very greasy drug molecule and get it into that solution can be quite nasty, and they have more side effects than the drugs themselves. So you can think of things that are good at solubilizing greasy molecules, these organic solvents like benzene and chloroform, and you probably know the names of these and know that these are not things we would want in the human body, and that's very true. There's other things like castor oil that can be effective at dispersing a drug, but we'll talk about why that's undesirable for other reasons. So we'll see that nanomaterials can be quite good at getting drugs that are difficult to work with into a better state. And then they'll also help us avoid some of the common side effects. Okay, so before we start talking about how we can use nanomaterials for anti-cancer drug delivery, let's look at a few examples of clinically used drugs and review some of the issues with using them as a treatment. And it turns out that there's just a few classes of drugs that are used for cancer chemotherapy. Some of these are molecules that react with DNA. And this is a good approach for an anti-cancer drug because this can shut down rapidly dividing cells because it deprives the cells of instructions for making more copies. Other drugs interact with the structural components of cells, and this also interferes with cell division. A third approach to cancer chemotherapy are molecules that act like natural metabolites, and they interfere with DNA synthesis. They shut down the enzymes that are responsible for copying DNA, and again, they deprive the cell of the instructions that they need to replicate further. So we'll look at a few examples of these. One of the anti-cancer drugs that's been around the longest is a molecule called cisplatin. And this is used to treat ovarian cancer, lung cancer, leukemia cancer, and quite a few others. The type of activity that it has is called DNA alkylation. Now, what does that mean? It's just kind of a dumb chemistry term, but maybe I can explain it. So this molecule sees a piece of DNA, and it sees the guanine residues within DNA, and it modifies them. It attaches the drug to them. And so now you have a piece of DNA that's there to be copied, right? It's carrying the genetic information, but it has this platinum molecule sticking off of it. And that really messes up the copying of DNA. So the alkylation just refers to the fact that we're hitting one of the functionalities on the DNA base and then interfering with its structure. So cisplatin has been around for quite a while. It was actually first characterized in 1845 when scientists were basically just messing around and looking at reactions occurring at platinum electrodes. They saw this interesting molecule being formed. And people kind of continued to play around with this platinum-based complex, and then somebody decided to look at it in the 1960s to see what it did to cellular function, and observed that it really stopped cells from replicating. So it was then put into a series of tests and through a clinical trial, and then it was approved as an anti-cancer agent by the FDA in 1978. Now, drugs like these, the DNA alkylators, affect both normal and cancerous cells. So they would definitely benefit from more specific delivery. Cisplatin has a range of side effects. It's really not a fun drug to be on. It causes kidney problems. It can cause hearing loss. It causes weight loss and nausea. And this type of drug is really dangerous to all cells. And so it's something that we would just like to minimize the exposure of healthy cells to. Another effective anti-cancer drug is doxorubicin. This is a drug that is used to treat certain leukemias and bladder cancer, breast cancer, tissue sarcomas, and lots of others. So doxorubicin works a bit differently. So it doesn't modify DNA, but it just slips in between the DNA base pairs. And what this does is it makes it difficult for cells to copy their DNA because there's this extra molecule between the bases. And then part of what needs to happen during the copying is that the two strands need to be pulled apart, and with this molecule in there, that's tough to do. So the DNA copying gets shut down, and then this helps to shut down cell division. So this is another way to interfere with the dividing of cancer cells. So doxorubicin, where did we find this molecule? It was actually originally isolated from a microbe that had a bright red color. And it's actually the color of a ruby, and that's where the name of the drug comes from with doxorubicin. The bug that produces it probably uses this molecule to fight off other bugs. So this molecule was originally thought of as an antibiotic, but then we figured out later on that it has this anti-neoplastic activity or anti-cancer activity. And so this class of molecules is sometimes called anti-neoplastic antibiotics. And I don't suggest that you try to say that 10 times fast, but just in case you ever hear the term, that's what it means. And it's interesting that organisms like bacteria bother to make these very complicated molecules, but you have to realize that bacteria live in a world of thousands of other kinds of bacteria. There's lots of bacterial warfare that goes on, and the way that they protect themselves from their neighbors is sometimes to have a molecule on hand that is harmful to somebody else, but not harmful to them. So many of these compounds have been found to be good at killing cancer cells while having some selectivity against healthy cells. But the main problem with using doxorubicin is that it causes damage to the heart and also to the lungs. And the cardiotoxicity that's seen after doxorubicin treatment is a major side effect, and it can actually be life-threatening once a certain dosage is reached. So we obviously need to find ways to keep doxorubicin out of the heart. Now, the second approach that I mentioned are drugs that interact with the structural components of a cell. Taxol, which is the name that it was originally given for reasons that I'll get to in a minute, the name of the drug when it's used clinically is actually paclitaxel. This is a drug that is widely used for the treatment of lung, ovarian, and breast cancer. And interesting enough, this is where the name comes from. It was originally isolated from the bark of a Pacific yew tree, Taxus brevifolia. And so that's where the name came from. Now, why were we looking at tree bark for drugs? That may seem like a pretty crazy thing to do. But it turns out that in the 1950s, the U.S. National Institute of Cancer started up an aggressive program to look for naturally occurring drugs. So we know that, for example, Native Americans, other populations that don't have access to what we call westernized medicine or modern medicine, very often rely on natural remedies. So they look at things around in their environment, they've kind of tried different things, they know what cures they may have out in their backyard for different ailments. And so there was a move by the U.S. government to try to collect some of the same information by studying the molecules in plants and in the environment. So there was a group of botanists that were recruited from the USDA who were sent out in the field, and they were told to gather as many samples as they could of different kinds of plants. Now, they would bring them back into the lab, they would extract all the molecules from the plant, and then they would see if it had any effect on either cultured cells, so cells that can just be grown in a flask in the lab, or on mice. So in the mid-1960s, an extract from the Pacific yew tree was tested, and very high levels of activity were observed. Now, this is a great finding, but it also presents a bit of a problem. So are we going to cut down every Pacific yew tree in the world to get this drug? No, obviously not. That's not a sustainable solution. So how did people contemplate taking this drug and moving forward with clinical testing? Well, it turns out that Taxol is a very complicated molecule. It has lots of bonds, and the bonds have specific orientations that nature is very good at programming. So nature is great at making complicated molecules like this one. For synthetic chemists, and they're the people that would be able to make large batches of the molecule, making this compound was quite a bit of work and very challenging. And eventually what happened is that pharmaceutical companies were basically able to marry the biology and the chemistry, get cells to do a lot of the work, and then they were able to produce the drug in a way that didn't involve cutting down any yew trees. So that was a very good solution. And this drug was eventually approved by the FDA in 1992, and Bristol Myers Squibb was the first company to offer it. The sales of this drug peaked in 2000. This was before there were any other analogs with the same activity available. And in a single year, $1.6 billion worth of this drug was being sold. It's now off patent, and it's available as a generic drug and is used widely in anti-cancer therapy. So how does Taxol work? It binds to a part of the cell that is quite important for structure, a part of the cell called the microtubules. The microtubules are actually nanoscale structures that are about 25 nanometers in diameter. They provide support for the cytoskeleton of the cell. So when the cell divides, the cytoskeleton actually needs to be disassembled. But when Taxol is bound, this is made much more difficult. The cytoskeleton can't be broken down to make way for new cells, so this is another way to shut down rapidly dividing cells. The major problem with using Taxol is that it is a large, greasy molecule. As I mentioned, it's a very complicated structure, and it's very hydrophobic. It doesn't like water, and that makes it very difficult to formulate. So it needs something really kind of greasy to make it stable in solution. So what was arrived on as a way to get it into solution is a form of castor oil. And again, it might seem like kind of a strange thing to test, but castor oil has been around for a long time. It doesn't taste very good, but it's at least a kind of a biological substance. But it turns out that many people react badly to the solvent. And I'm not talking here about the bad taste wanting to spit it out, but instead that castor oil, when it's used as an intravenous injection, can cause certain people to experience neurotoxicity. So clearly we need better ways to deliver drugs like Taxol. So now you have a better appreciation for why we need better solutions for drug delivery. We need to keep drugs out of the sites in the body where they can be harmful, and we need to be able to introduce them into the body without carriers that produce side effects of their own. So let's use what we know about the world of nanoscience to survey the range of approaches that are being explored to solve the problems of drug delivery. So one of the first classes of materials that we can look at are carbon nanotubes. if you think about the structure of a carbon nanotube, you have this very hydrophobic shell of carbon and then an empty space in the middle. So that almost looks like a drug capsule, doesn't it? And the fact that carbon nanotubes are very hydrophobic means that these drug molecules that I've talked about as being very problematic to formulate will be happy inside of a carbon nanotube. We've also talked about the fact that carbon nanotubes can be biodegraded. So they can be attacked by enzymes like peroxidase, and that allows them to be broken down in the body. So that's a very useful property that carbon nanotubes have. It also turns out, and this is a very interesting development, that people have learned how to cork carbon nanotubes. So if you think about the way the structure usually appears, it's an open-ended capsule. That wouldn't be all that useful, right? If you just have an open capsule floating around, let's say in the bloodstream, it's eventually going to spill the drug into the blood. But people have figured out how to kind of snip the ends shut, but then have them opened back up once they're in the right environment. So that's a very nice type of switchability that allows these materials to be used with quite a bit of flexibility. And another neat thing, and we'll come back to this in the next lecture, is that you can actually coat carbon nanotubes with drug on the outside. So rather than putting it on the inside, you can layer a bit of drug on the outside of a carbon nanotube, and that then gives you kind of a finite dosage carried on that nanomaterial. And what we have here is also a picture of a very neat kind of carbon nanotube that's actually a carbon nanohorn. So this is a type of carbon nanotube which has solved the open capsule problem by actually growing one side of the carbon nanotube into a horn shape. And you'll see in this image that there's actually cisplatin particles in this nanohorn, and they're sitting there waiting to be released once they get into a tumor. So now let's talk a bit about liposomes. This is another major class of carriers for drugs that are nanoscale. Now, we just looked at carbon nanotubes. Those are kind of a hard nanomaterial. Their structure is really fixed. Liposomes are different. Liposomes are really soft materials. And they're mainly made out of the lipids that make up all of our cells, that make the membrane around all of our cells. So what happens here is that we formulate a drug by putting it in the middle of a liposome, and then we form the lipid bilayer around the drug. And so you have this capsule of drug, lots of drug molecules inside, encapsulated by something that the cell is very used to seeing. So again, excellent biodegradability here. And so we'll come back to this in the next lecture with some very specific examples, but these liposomal carriers have turned out to be very powerful for getting around some of the side effects that we see with drugs that don't have any kind of nanopackaging. Now, another class of nanomaterial-based drug delivery agents that we'll look at are protein nanoparticles. So protein nanoparticles are a great vehicle for drugs because not only are they biodegradable, but the cells recognize them as just being proteins. So if you can find a way to make a particle out of protein, stuff it with drug, this is a great way to move a drug around the body and get it to tumors very specifically. So another thing that we'll find that nanomaterials can do is get around some of the resistance mechanisms in cells. So drug resistance and drug resistance to anti-cancer agents is a major problem. We have people that start responding very well to a therapy, and then after a few dosages, their tumors become resistant. And this is because cancer cells are very aggressive, and they're very good at coming up with ways to get rid of a drug so they can pump it out of the cell. They can overexpress that pump in a very short amount of time. They can also introduce enzymes that will deactivate a drug. But if you package the drug up with a nanomaterial, that really fools the cell into thinking that it's not there. And if you can have the action of the drug be much faster, you give the cells much less time to become resistant. So that's another advantage that we'll revisit of packaging drugs into nanoscale delivery vehicles. And there's some other very interesting things that are just being developed for drug delivery based on nanomaterials. There are things that are referred to as nanovalves. So these are drug packages that can come up to a cell and they will sense the pH of that cell. So the pH tells us whether something is acidic or basic. It turns out that healthy cells have a different pH than cancer cells. So these nanovalves can come up to a cell, they can figure out what pH it is, and if it has the pH of a cancer cell, only then will these things release the drug into the cell. So that's very specific, very powerful. Now, we've just gone through a survey of different nanomaterials and how they can be used for drug delivery. How do we study this? How do we take a drug, package it up, and then figure out whether we've done anything to improve it? So there's lots of ways to do this, but the most popular way is to use cultured cells. So we have cells in flasks that we can grow in the lab, and we can introduce the drug, always do controls to make sure we're not looking at an artifact, and we can look at the effect of the drug kind of with and without the nanomaterial-based carrier and see if there's an increase in activity. Another way that we can look at drug efficacy, of course, is to use animals. So once you have some proof of principle, some experiment that you've done with cells, you can then move into an animal to see if you can reproduce that same effect. But here where you have the circulatory system to contend with and all of the other intricacies of a live animal. Another way that we can study drug delivery that's very unique to using nanomaterials as drug delivery agents is that we can use things like scanning electron microscopy. So if we have a particle that we can see, we can use that as a carrier, and then we can image either the cells that we're testing or even an animal and figure out exactly where the particles went. See if they got into the cells, see where they went within the cells, and really get the full profile for what's happening at a very fine level. So now we've looked at many of the key concepts that are important to appreciate so that we understand the challenges and solutions that exist in the area of drug delivery. We've considered how conventional drugs are distributed in the body and what the issues are with the conventional approaches. We've also worked our way through the major classes of drugs that are used to treat cancer. And we've looked at the major classes of materials that can be used to solve the delivery problem. So with all this information covered, we have all of the pieces, both the available drugs and the available delivery mechanisms, so that now we're ready to turn in the next lecture to how this all comes together. And in the next lecture, we'll look at examples of nanoscale drug delivery systems that are already playing an important role in cancer treatment. And we'll also look at examples of some newer systems that are in very early stages of development that are quite promising. See you then.